Hello and welcome to today's webinar. My name is Ann Holmberg and I will talk to you for the next 30 minutes regarding Gene Set Enrichment Analysis. So let's start with that. And if you have any questions, please use the chat. I will monitor the chat and try to answer questions as they come along. And of course, there will be questions at the end of this presentation as well. So the common approach to gene set enrichment analysis is that you order genes according to their differential expression between the groups that you are interested in. And then you focus on the most significant genes and you try to find biological clues, typically enriched pathways. And even though this is called gene set enrichment analysis, it's not limited to genes. It could really be anything, proteins, metabolites, and so on. The trick is that the pathways that you compare with must have the same unique identifier as in your data set. And the most common pathways have gene symbol. And the GSEA method was invented in 2005, and this is the original paper on that by Subramanian et al. And it works on gene sets, or which is typically pathways, and they are created based on biological knowledge. And then you rank the genes in your data set with a suitable metric that mimics what you're interested in. So standard statistical methods like Ttest, Ttest, and so on you use there. And then you calculate the enrichment score for each gene set. And the enrichment score tells you the degree to which this gene set is overrepresented in the top of your list or in the bottom of your list, representing the upregulated or downregulated variables. And then we also compute the P and Q values for the gene sets to tell you if what you have found is statistically significant. So that's also very important. And before you start, you need to prepare your data set. So it's important that we have the correct identifier and that it's unique. So if this is one of the pathways that you are comparing with, you see that here we have gene symbol as the unique identifier. Let me just kill my video as well, so that. And here is my data set, let's say that. And then we see that in my data set, I have a probe set ID as the unique identifier, but I also have the gene symbol information in there. So in this case, what I need to do is to make the gene symbols a unique identifier. And then I must make sure that I only have one value per gene. Here you can see that for this gene, MOSPD2, there are 2 values. So I need to create an average so that I only have 1 value. So that's very important. So the correct identifier, and it needs to be unique. So I will show you how we do that in Qlucore when I do the demo. And then once we have matching symbols, what we do then is that I rank my list, my data set. So I have my most interesting gene. If it's a Ttest, I will have the most interesting upregulated genes in the top of my list and the most interesting downregulated genes in the bottom of my list. And then I step through my data set gene by gene and I compare with each of the pathways. So here you can see that the first gene in my data set is present in the pathway. The next one is also present. The the third one here is not present, this one is present, and so on. And while doing this, I'm calculating the running enrichment score, which I will show you in the next slide. So here you see every time I get a match with the pathway that I'm comparing with, I take one step up, and every time I don't get a match, I take a step down. And this then here generates a maximum. So here I have a lot of matches, which means that I step up, and then I have fewer matches here, so then I go down. So the maximum here is the enrichment score, the running enrichment score. And you can see down here, each blue line is a matching gene with this pathway that I'm comparing with. And I have a lot of matches in the beginning here. And that's why I get a positive maximum here. So I step through my list in this direction, like that. And here we see, so the question is, do the members of the pathway, are they randomly distributed over my gene set or over my data set, or are they enriched in the top or the bottom of the list? In this case here, we can see that they are enriched in the top of my list. Down here, we see that here, since we're doing Ttest, we have a positive metric here, the T statistic, and we have a negative T statistic here. So most of the genes are found here in the beginning of my list, meaning that they are upregulated. And here in the list view, we can see that these are the core enriched genes. Okay, so I need gene sets to compare with, and gene sets or pathways can be downloaded from various sources. One very common one is MSigDB, Molecular Signature Database from Broad Institute. You can also find gene sets at Reactome, Gene Ontology Consortium, and so on. So there are many sources for this. And a gene set is very often a GMT file. And that means that you can have several pathways per file. And the format then is that you have one pathway per row in this file. So in the first column, you have the name of the pathway. In the next one, you have the description. And then you have one gene per cell or per column here. So here you can have a large number of pathways in one file, a GMT file. And GMT, it's basically a text file that has just been renamed to GMT. So it can easily be opened in Excel, for instance. So I see I got a question, so let's have a look at that. Yes, good question. Are these gene sets required to come from the same species? Yes, that's very important, because the gene names are, of course, species specific. So you cannot compare a mouse data set with human with a human pathway. But you can, but you need to convert then your mouse genes into human autologs. So I will talk a little bit more about that later, but it's definitely species specific. You can also use TXT files when you have one set per file, and then it's a list going downwards. And these TXT files can be lists that you have generated from our software. And then you need to select which test that you want to use to rank the genes in your data set. And in GSEA, the genes rank differently depending on which test that you use. And we rank according to the test statistics. And for ANOVA or F-statistics, or multi-group comparison, as we call it in Qlucore, the test statistic value is always positive. In a T-test, or a two-group comparison that we call it, the test statistic value, the T-statistic, may be positive or negative depending on if the genes are upregulated or downregulated. And you can also rank based on other statistical tests. For categorical predictors, you can use Ttest or ANOVA, and if it's numerical, then regression testing is used for ranking. Okay, so that was a little introduction, and now I will show you what it looks like. So here we are. So I will. So any more questions before I start the demo? Nope. Then we continue. So I opened up the software, and then I will load one of the example data sets. So the big one, the acute lymphoblastic leukemia data set, where we have 132 samples and 22,000 genes or variables. And in this data set, I will show you so you understand what we are looking for. In this data set, we have identified which subgroups of leukemia that the patient suffer from, or not we, but actually the author of this data set or at the hospital where they were treated. So this is annotations in this data set. So we know that there are different subtypes. And what we are interested here is a separation of one specific, the TALL. And we know that it's very easy to find a lot of statistically significant genes that are good at separating this group from the rest of the samples, as you can see. So now I've filtered down to around the 1900 best variables. I don't need to do this before I start the Gene Set Enrichment Analysis, so this was just to make you understand what we are looking for. So these are the genes that we are trying to find enriched pathways for. And before I start the Gene Set Enrichment Analysis, I must make sure that I have the same unique identifier in my data set as in the gene sets that I'm going to compare with. And we can see that in this data set, it's probe set ID. And I want to compare with gene symbols. So I need to go to the Data tab and change the identifier here. And I do have gene symbol in my data set, so I just change to make that the unique identifier. But the problem now is, if we look here, that I have 13,000 unique genes, but I still have 22,000 data points. And that's because I have several probes that refer to the same gene. So I must now create a unique value per gene. And the way to do that is to collapse. Still in the Data tab, I collapse, and I have 4 different ways of doing that. Pick the maximum, the minimum, the average, or the median value. I pick the average. So now the average value for each gene is calculated, and now you see I have the same number of genes as the same number of unique values. Good, then I'm ready to go. Then I open the Gene Set Enrichment Analysis workbench here, and it is loading there. Here we are. And here, as you can see, I can see which is the data set, how many samples, how many variables, and what's the unique identifier. So that's good to check that I have the right thing here. And even if I did a statistical filtration before I open that, that will not have any impact. I will still use all my variables. But since I ranked them, I will still only take the most statistically significant into consideration. And here I have 3 gene sets that I can use to compare with, These gene sets are shipped as examples with the software, and we don't update them regularly. So you should not use these. Instead, you should download your own gene sets from public sources, or maybe if you created them yourself. And then you put them in a folder on your computer, and then here you point to that folder, and then they will show up here. And then you can select one or many here. So I select now the reactome pathway subset, which contains more than 1,000 human pathways. And then we go down here, and here I must select the metric that I want to use. So it picked the one that I used in my statistical dialogue, and that's what I want to use. As you can see, I have all the other standard tests available, plus the signal to noise ratio. But since I'm interested in the genes that are good at separating this group from the rest of the subtypes, Ttest is a good ranking method here. So that's good. If we go here, I have some settings where I can set the minimum match set size. So if you have very small gene sets or few genes in your data set, maybe you might want to make this number, set it to 1 or something like that, or even 0 maybe. Otherwise, we won't show pathways that have a smaller number of hits than this. And there's also a maximum value. Here is the permutations. So we will permute 1,000 times on the sample side in order to calculate the P and Q values. So normally you do it on the sample side. If you have very few samples, you can also do it on the variable side. Yeah. Okay, and then we are ready to run. So I just click run here, like that. So now I am comparing each of the gene in my data set, each of the genes in my data set with each of the pathways. And I'm permuting this 1,000 times. So it's a lot of calculations going on. And here we already have the results. So this is done very, very fast. And here we can see that cholesterol biosynthesis comes up high in the list here with a good enrichment score and a good Q value. The P and Q values are calculated in a different way than from what you are normally used to in your normal statistical analysis, because here we do permutations. So a Q value of up to 0.25 is considered quite okay in the GSEA. So here we have a good Q value, and we see here is the running enrichment score. We see that we have a positive maximum because we have a number of enriched genes here in the beginning of my data set, meaning that they are upregulated, since this is a Ttest. And the maximum value here is the same as this one, the enrichment score. And then we also normalize that. So that's a normalized enrichment score. If I go down to the bottom of the list, you see that I find a list with good Q values here. Here, for instance, the interferon gamma signaling. And if I click on that one, you see that we have a negative maximum, meaning that it's mostly downregulated genes that are enriched. And if I now sort on Q value, like that, and I can filter on Q value, say I can accept anything below, say 0.15, then I find 5. And for each of them, I have the the enrichment score, heat map showing the matching genes, how they are expressed in my dataset, and a detailed list of the genes, which shows which are the enriched ones. And if I mark all my 5 lists here, like that, and pick the heat map, I will get a leading edge overlap here. So that helps me see if any of these pathways are overlapping. And you can see that 2 of them actually are. So CDO in myogenesis and myogenesis seems to be identical, actually. You can see they both contain 29 genes and have 28 matches. So this is probably the same pathway with 2 names or something like that. Okay, and if I would like now to export all this information, I just press results here and I put it somewhere where I can easily find it. So I put it on my desktop. And here you see that now I have exported everything. So here I have all the heat maps, including the leading edge overlap. Here I have all the result lists, all the enrichment scores, so on. So by just pressing one button, you export everything in one go, like that. And if I press lists here, like that, these lists have now been exported back to my normal working environment. So they are actually now here among my variable lists. And that's really interesting because now I can actually use these lists to explore further and understand more. So let's again filter down to some of the best genes here. So I filter down to around the 900,000 best. Here you see I have fantastic Q values, so they are highly significant. Then I open up a new synchronized plot and I make this a variable PCA plot like this. And when I do a t-test, the variables are always distributed in 2 groups. And these plots are synchronized, and that means that genes or variables over here, they are upregulated for samples over here. And genes over here, they are downregulated for these samples and upregulated for some of these samples. So I can show that by just coloring on how these genes are expressed for my group of interest, which is the leukemia subtype TALL. And then you see that they are all upregulated here and all downregulated here. If I would color according to something else, like the MLLL, you see that I have some slight upregulation here and downregulation. So then it looks totally different. But now I want to use these pathways. So now I click this little box here, and that means that I color my genes according to this pathway. So all genes that belongs to the cholesterol biosynthesis pathways are colored yellow. So you see 2, 1 here, so they are a bit hidden. But by making this list the input, then you see that it's easier to see. So I have 7 genes among my best 900 that belongs to the cholesterol biosynthesis pathway. And I can use the label tool to label them like that. If I would like to save this, I can just make a copy of the active list, which now contains these 7 genes, and copy and say these are cholesterol biosynthesis separating TALL, like that. And then I take that away to get back and see everything. And then I can do the same for interferon gamma signaling. And then you see that the pink ones belong to this pathway. And they are now mostly in the downregulated group, and that makes sense because here we had a negative enrichment score, so we expect to find downregulated genes. And to show only them, we can do that, and then we see that most of them are downregulated, but it seems like I also have an upregulated one. And again, I can make a copy, or the other way is to just make an empty list here. Use the list tool and then circle everything or the ones that you want, like that. So that's another way of populating a list. And then we can just rename it to interferon gamma signaling separating TALL, like that. So you see that we have a a seamless interface with the GSEA workbench. So you can not only identify pathways, but you can actually bring them back and work with them and use them to further understand what it is that you have found. So that's very, very powerful. So that was the end of the demo, and now I will talk a little bit more. Any questions on the actual demo? No? Then we continue. So let's just maximize this again, like that. I would like to say a few more words regarding the ranking. So the Ttest ranking is useful if you have a pathway where you expect all genes to be either upregulated or downregulated. Because then it will be easy to identify either positively upregulated or downregulated, either in the top of the list or at the bottom of the list. So if you rank with ANOVA or Ttest or multi-group comparison, this can be useful if you have a pathway where you expect to find enrichment, but you are not sure if all of them are in the same direction or if they are maybe mixed. So then ANOVA can be very useful. So let's go to then. So what should you use then? So you cannot generally say which ranking method to use. You need to have an idea of what you expect in your data set. Because the thing is that you will see a big difference in the P and Q value between the use of an Ttest, ANOVA, and a Ttest in the GSEA with the same groups in the ranking test. In the normal statistics, there is no difference if you have two groups and do a multi-group comparison or a Ttest, but it is a big difference in the Gene Set Enrichment Analysis. So that's important to remember. So if you have a large number of pathways and you don't really know what to expect, you can always test with both Ttest and ANOVA and have a look at the outcome. And then you can go back to the result and see what makes most sense with your data set. So there are no firm rules here on what to use. And also a little advice on the question that I got before. So if you working with species other than human, what should you do? Yeah, so most of the pathways that you find are based on human gene signals, and then you need to convert your data set. And there are a number of ways to do that. So here is 4 different ways of doing that. And I will send you these slides. You will have this information. Yeah, I think that was actually it. Now, 30 minutes later. So do you have any questions? Please ask them in the chat. While you are thinking about that, I can just let you know that there are more webinars that you are very welcome to attend. So we run 3 next week on different import of different data type or analysis of different data types here, and a little bit about exploration. The next basic training is the 24th of February. And you know where to register, and you know where to download a free trial. Or maybe if you didn't download a free trial, please go to our homepage and do that. You have a 10-day free trial if you want to try the software. I got a question if I can go through my first 3 slides again. Yeah, I can do that if you hang on a little bit. And if you have any questions, you are very welcome to contact either me directly, or if you're already in contact with my colleague Jan, you can contact him, or of course our support. So if there are no more questions, I would like to thank you very much for your attention today. And when you close this webinar, you will find a small poll. So if you have time to give us some feedback, that's always very appreciated. So thank you very much and have a nice day. Bye-bye. Oops. And now I will show the 3 first slides again for if somebody came in a little bit late. Okay, so the common approach to gene set enrichment analysis is that you order genes according to the differential expression between the groups that you're interested in. And then you focus on the most significant genes and try to find biological clues, typically enriched pathways. And the gene set enrichment analysis method was invented in 2005 by Subramanian et al. And this is the original publication on that. And it works on gene sets or pathways that are created based on biological knowledge. And then you rank your data set by a suitable metric. And then you compute the enrichment score for each gene set, and the enrichment score tells you if you have an overrepresentation in the top or the bottom of the ranked list of the genes in your data set. And then you also compute the P and Q values to establish if your results are statistically significant. Okay, so did you see this set array, or do you want me to continue? Okay, so let's have a look at this as well. Okay, so when preparing your dataset, you must first check your pathway to see what is the unique identifier, because you must have the same identifier in the pathway that you're comparing with as is in your dataset. So in this case, the pathway has gene symbols as their unique identifier. And in this case, my dataset has probe set ID as the unique identifier. So, but I also have gene symbol as one of the annotations in my dataset. So I then change to that, and I make sure that I have unique values for each gene, which I showed in the demo. And then you start a comparison. So you check each gene in your data set with each of the pathways to see if they match. So in this case, I have a match in the pathway for this gene as well. And then for the next one, it's not a match, and so on, and it is a match. So the question is, so you ignore the probes with the same gene symbol? No, I don't ignore them, but I create a, as I showed in the demo, I create, for instance, an average. So I calculate the average for the 2 probes or 3 probes with the same gene, or I take the maximum value or the minimum value. So I create one value per gene, and I can do that in a number of different ways. And that's what I showed in the software here, that I collapsed the data here. So here I changed to gene symbol, and then I collapsed on average. You see I have 4 different ways of collapsing, meaning that I only have one value per gene in this case. Okay. No, question is, do you recommend average? No, I don't recommend everything, anything. So that's your choice. So that's why we have four choices, 4 options. And this webinar is also recorded if you want to, so it will be published in a couple of days and available on our homepage if you want to go back to it. Great, thank you. Thank you for your time and have a nice day. Bye-bye.